Hello, now I will present to you a little bit about the biology of HIV and AIDS, and it will be presented in 3 sections. And the first one is about the 9 genes of a killer. The theme of the whole section on the biology of HIV and AIDS is that, first of all, the HIV is, though we know just about everything about it, it's very difficult to cure, to treat, as a matter of fact, and certainly to prevent. HIV is a virus, and drugs in general cannot kill viruses. The other problem is that HIV lives in and at the same time hides from the immune system. And later on, I'll explain how that works. So we know everything about it, but we can't cure it. And these 3 facts are going to serve as the theme throughout the 3 different sections of the biology of AIDS. So the learning objectives for the first part of the 9 genes of a killer is that you would be able to define what a virus is, that you could distinguish HIV, which is a retrovirus, from other viruses, that you would be able to describe the major structural components of HIV, also that you can relate the HIV life cycle with how it takes over the host cell, and then compare the HIV genome with that of other organisms. So first of all, what is a virus? It's different than other microorganisms that infect us. Viruses are extremely small. They are so small that you can't even see them with a light microscope. Even the most strong lens of a light microscope does not distinguish a virus. Also, all viruses require a host cell to both live in and reproduce. They cannot live on their own. Even bacteria are able to live on their own without a host cell, but viruses cannot. Now, a virus can have its genetic material expressed 2 ways, either as DNA or RNA, and I will speak about these shortly. So HIV is a retrovirus, and we call it a retrovirus because its genetic material is coded as RNA. Most organisms' genetic material is encoded as DNA. And in order to understand why this matters and what the difference is, we have to first describe what DNA does and what RNA does. So DNA actually has 2 functions, and one of its major functions is to replicate itself, make a copy of itself. And the second major function of DNA is to transcribe itself into RNA. By transcribe, we mean to rewrite itself as RNA. Now, the function of RNA is simply to translate those genes, that genetic code, into a protein. So the reason I'm even talking about this, it becomes very important in understanding HIV's life cycle, its infectivity, and so forth, because HIV must first do what we call reverse transcribe. That means rewrite itself back into DNA and then again into RNA. And that's why we call it a retrovirus. It has to commandeer our cells to help it rewrite its RNA into DNA. And you will see why this is so important. So to understand this, let's take a little look at the actual structure of the virus. And it's actually a very simple structure, and it has few proteins and few parts to it. If you want to look at the outermost part of the HIV virus, it's surrounded by a lipid membrane. And this lipid membrane is actually your membrane or my membrane. It actually borrows this from our cells. And you will see how that plays into the HIV life cycle as well. It's very important. Inside of the virus is a capsid, and inside of that little capsid, in the little diagram, the capsid is surrounded by orange colored proteins in this little cartoon. And inside of that capsid is its RNA, is its genetic material encoded as RNA. And one virus has 2 strands of RNA. So other important proteins, if you look at the slide, are depicted by the little green stalks coming out of the lipid membrane. And those consist of 2 proteins called GP120 and GP41. And those become really important when we talk about the infectivity of HIV and the HIV life cycle. And they, as you will see, they serve as the site where they actually attach to the host cell. So this slide shows you the HIV life cycle, and it shows you the entire life cycle in one cell because that's where it occurs. And if you follow along on the numbers, that it will show you what happens in sequence. So the first thing is that the HIV virus must find its cell that it serves as its home. And the second part of this biology of AIDS, we'll talk about that cell, but for right now, the most common cell that the HIV lives in is the lymphocyte. So the particular lymphocyte is called a CD4 lymphocyte. And so what the HIV is essentially looking for is that CD4 marker on the lymphocyte, because it is that CD4 marker that it finds and links up with via that very important GP120 protein. So the GP120 and the CD4 actually act as what we call a receptor and ligand, and they hook up together. And now the HIV knows that it has the right cell. We will talk later about what the function of this CD4 is for the lymphocyte, but for now, the function of it for the HIV is to attach, and then it utilizes a second protein on the lymphocyte surface as what we call a co-receptor, and we will talk about that later also. So one of the co-receptors is called CCR5. So via these 2 lymphocyte proteins, the HIV attaches, and then it comes close to the host cell surface and actually fuses with it. And this is the important moment, because remember I said that the membrane of the HIV is your own lipid membrane. So when they fuse, they become as one. And what this allows now is for the virus to release its contents into the host cell without interrupting that cell at all. It's as if nothing happened. So lots of times people think, well, a virus sort of bores into a cell or forces its way in. But in essence, the virus simply attaches to, and the cell literally says, welcome, come in. So what happens now, and if you look at number 2 of the slide, on the slide of the HIV cycle, you'll see that a few of the proteins and the genetic material, of course in the form of RNA, is now released into the host cell. Things happen very quickly from here, because what happens now is that some of the proteins of the HIV, some of those enzymes kick in, and first of all, they allow the RNA to be quickly rewritten as DNA. And we call this reverse transcriptase, this enzyme. And this becomes key also later on when we start talking about the drugs. So this reverse transcriptase rewrites the RNA into DNA. And why that becomes so important is that now this new DNA, this viral DNA, actually incorporates into your own DNA in your nucleus. So it becomes as if it's part of your own DNA. Now, at this point, several things could happen. If the virus stays sort of dormant, the cell will actually stay in this form, keeping the viral DNA as part of its own DNA, and nothing in particular could happen. Or the HIV will go on to complete its life cycle. And what that entails now is to command that bit of DNA carrying those viral genes to transcribe into RNA for new HIV viruses. And also then the RNA gets what we call translated into protein. So those two things have to happen before more HIV viruses are made. And when they do happen, they assemble together with a few proteins that the RNA commanded to be formed, and they assemble near your cell surface or the host cell surface. They then become encompassed by new lipid membrane, once again from the host cell. And when this happens, they actually can bud off of the surface of the cell. So if you'd like to see this sort of animated and in a continuous motion, if you would refer to the YouTube, then that's about a 2-minute little presentation of the HIV life cycle. So finally, it's worth mentioning about the genes themselves. It's amazing that HIV has only 9 genes. And compare this to a simple organism like E. coli, which is a small bacteria that lives in your intestines, that has 4,000 protein encoding genes. Or a human being has 20,000 protein encoding genes. The HIV has only 9 genes. Very simple, but these genes are very powerful. So this slide of the HIV genome actually gives the names of these 9 genes. It gives the function of the genes, and it describes a little bit about the function. So we essentially know the entire genome. We know exactly what each gene does. We don't know that about our genes. And this also makes it all the more remarkable why we seem to have such a hard time treating and preventing the virus. So in summary of this first part, HIV is a retrovirus, meaning that its genetic information is in the form of RNA and has to be rewritten as DNA before it can replicate. It must enter a host cell and use the host cell to replicate. It can never do this on its own. And finally, HIV has only 9 genes, whereas E. coli has 4,000 and the human has 20,000. And these 9 genes are able to make all the proteins necessary to complete that HIV life cycle.